What is DNA? DNA, or deoxyribonucleic acid, is a molecule composed of two polynucleotide chains carrying genetic instructions for the development, functioning, growth and reproduction of all known organisms and many viruses. Alongside proteins, lipids and complex carbohydrates, nucleic acids are one of the four major types of macromolecules that are essential for all known forms of life. To understand how DNA sequencing works, we first need to understand its structure. The two DNA strands that coil around each other to form a double helix are known as polynucleotides, as they are composed of simpler monomeric units called nucleotides. Each nucleotide is composed of one of four nitrogen-containing nucleobases: cytosine, guanine, adenine or thymine, a sugar called deoxyribose, and a phosphate group. The nucleotides are joined to one another in a chain by covalent bonds between the sugar of one nucleotide and the phosphate, resulting in an alternating sugar-phosphate backbone. It is the sequence of the four nucleobases along the backbone that encodes genetic information. Now, looking into our topic, what exactly is DNA sequencing? DNA sequencing is determining the order of the four nucleobases. The sequence tells scientists the kind of genetic information that is carried in a particular DNA segment. For example, the sequence information can be used to determine which stretches of DNA contain genes and which stretches carry regulatory instructions, turning genes on or off. How is DNA sequencing done? Regions of DNA up to about 900 base pairs in length are routinely sequenced using a method called Sanger sequencing, or the chain termination method. Sanger sequencing was developed by the British biochemist Fred Sanger and his colleagues in 1977. The chain termination method requires a DNA template, a DNA primer, a DNA polymerase, normal deoxynucleotide triphosphates and modified D-deoxynucleotide triphosphates, the latter of which terminate DNA strand elongation. The D-deoxynucleotides may be radioactively or fluorescently labeled for detection in automated sequencing machines. The DNA sample is divided into four separate sequencing reactions, containing all four of the standard deoxynucleotides and the DNA polymerase. To each reaction is added only one of the four dideoxynucleotides, while the other added nucleotides are ordinary ones. The mixture is first heated to separate the strands of the template DNA, then cooled for the primer to bind. After the primer is bound, the temperature is raised again, allowing DNA polymerase to synthesize new DNA starting from the primer. DNA polymerase will continue adding nucleotides to the chain until it happens to add a dideoxynucleotide instead of a normal one. At that point, no further nucleotides can be added, so the strand will end with the dideoxynucleotide. This process is repeated in a number of cycles for all the four nucleotides. By the end of all the cycles, the tube will contain fragments ending at each of the nucleotide positions in the original DNA. The DNA bands may then be visualized by autoradiography or UV light, and the DNA sequence can be directly read. And that is how DNA sequencing is done.